Assalamualaikum and hi to everyone and Dr. Ng. Today, our group 5, will be presenting about the eyes open and eyes closed signal from EEG. So I'll start this presentation with the introduction of EEG. So EEG is a medical or biomedical instrument that detects and records the electrical activity in the human brain. So the waveform observed are supposed to indicate the activity of the cortex, which is the brain surface, and this electrical activity from the brain areas underneath the cortex, which can influence this activity. So the action potentials are actually signals produced from the nerve cells in the brain. So these action potentials actually go from one cell to another cell via a gap, which is known as a synapse. So the neurotransmitter is actually special molecules that aid in the signal transmission across this gap. And the brain generally works very hard to maintain an equal level of each of these neurotransmitters. So EEG activity is recorded in microvolts and with the primary frequencies of interest ranging up to about 30 Hz. So these are the electrodes of the EEG. So electrodes are actually little metal discs which we insert it in the specific locations on the scalp. So the record discs will identify these places by measuring the head using the intentional 1/20 system. So actually this is accomplished by measuring by collecting the measurements between the preset spots on the scalp, and the electrodes will be inserted at the side 10% and 20% of these length parts. So each channel actually trace an activity on the EEG machine. So this can be produced by the differential amplifier in the machine. So actually there are two inputs on each amplifier and each input is wired with an electrode. So the differential amplifier will measure the difference in voltage between these two signals at each of these inputs. So the resultant data is actually amplified and shown as an EEG activity channel. So the signals that are captured by the EEG can be mistaken as artifacts. So the signals that captured by EEG but not created by the brain, we can call it as artifacts. So some artifacts can be mistaken for their actual epileptiform abnormalities or seizures. So it is very critical to separate the artifacts from the brain wave. So we can separate it by understanding the logical topographic field of distribution for the actual EEG abnormalities. Now I'll pass to Daniel to further explain about the methodology of this assignment.
My name is Daniel Haikal. Today I'm going to talk about the methodology in this report. First of all, we need to plot the raw data. So how we do it? We import the raw data of EEG for eyes closed and eyes open and import in MATLAB. Second, we need to find the sampling rate. So first we need to do the assumption, which is 256 Hz, and then we need to convert the data into frequency domain. So based on the frequency domain, we can know the value of the peak. When we know the value of the peak, we can know the exact value of frequency. So after I test with 256 Hz, I can get 50 Hz of artifact noise. So is it true because the electrical noise in Malaysia is around 50 Hz? So it's true to use 256 as a sampling rate. And after that, we filter the signal, and the most appropriate filter for the signal filtering is the bandpass filter with frequency of 8 until 13 Hz. This is because in the alpha wave frequency.
Hi, so for my part, I will explain about the raw data of the EEG. In the figure 1, which is the table above, it shows the raw data of the eye closed without EMG, while the table below, which is figure 2, shows the signal with the EMG data. Both figures shows that the signal has a lot of artifacts, which really can affect the quality of the signal. For the figure 1, the artifacts may come from the EEG machine itself, or we can call it as the instrument artifacts. For figure 2, it shows more artifacts than the figure 1 because it has some additional artifacts from the muscle of the forehead, jaw, and the eyelid, as this muscle movement can cause artifacts by moving the electrodes itself. Movements in the surrounding produce disturbance by altering the ambient electrical fields. Moreover, the tongue and the eyes have their own dipole electric charge. Therefore, their movement can get recorded by the electrodes. Moreover, the movements and the blinks represent a major source of the artifacts in the EEG and event-related brain potentials. The origin of this artifact is the large difference in potential that exists between the cornea and the retina.
Now I will continue presenting and explaining about the frequency domain of eyes closed of the raw data. So in order for us to know the artifact of the signal, we need to convert the raw data into the frequency domain like shown in this figure. So the function of this frequency domain is actually to know the exact frequency at which peaks are occurring. So the frequency domain representation of a signals allows us to observe several characteristics of the signal that are either not easy to see or it is not visible at all when you look at the signal in the time domain. So there are also other methods to know the frequency of the peak, which is by using the FFT function. But in this report, we only show the method of using the spectral function. So we can see that each signal actually has different peaks, and from this figure, we can see that the highest peak is the blue one, which indicates the C3 of the central region.
Now we move to the filtering process, but before that, as I said before, the frequency range of this filter is from 8 until 13 Hz. This is because this range is alpha wave, because the alpha wave is the most dominant brainwave that is likely to be observed in EEG data. Okay, now we filter the raw data of eyes closed in one figure. As you can see, there is the difference of wave pattern before and after filter. Before filter, the amplitude tend to be higher rather than after filter because there is filter of the noise. But there is some noise that we can't filter because based on the research that some of artifacts that are impossible to filter using the bandpass, we need to use other tools that can filter the noise like ICC, BEG, and so on. Okay, next. We filter the raw data of each electrode. So we have C3, C4, O1, and O2. For this graph, you can see clearly the difference amplitude before and after filter for each electrode. For before filter, the amplitude is different for each frequency, but when filter, the amplitude is quite similar for each frequency. Okay, next we filter the spectral function. But before that, there are two types of frequency domain, which is FFT and spectral. In this report, we use the spectral function. Okay, so we filter the spectral function of eyes closed data, and we see the result of the filter. Before filter, we can see the highest peak is come from blue color, which is the C3. But when after filter, the highest peak is come from yellow color, which is the O1, the occipital region.
Next, let's talk about the EEG data, the raw data from eyes open. Figure 8, which is the table above, shows the raw data for the eye open without the EMG, while figure 9, which is the table below it, shows the signal with the EMG data. Both figures shows that the signal has a lot of artifacts, which can really affect the quality of the signal. For figure 8, the artifacts may come from the EEG machine, or it can also be called as instrument artifacts. The artifacts may come from the stimulation of the brain due to stimuli seen by the subject in the experiment room, such as distraction from a moving clock, or it has some movement from the other group members. For figure 9, which is the table below, it shows more artifacts than the figure 9 or the figure 8 because it has some additional artifacts such as the muscle from the forehead, jaw, or the eyelid muscle, as this muscle movement can cause the artifacts by the moving of the electrodes. Furthermore, the movement of the eyelid may increase as the subject can see what is currently happening in the experiment room. Movement in the surrounding produce disturbance by altering the ambient electrical fields. Moreover, the tongue and the eyes have their own dipole electrical charge. Therefore, their movement can get recorded by the electrodes. Moreover, the movement and blinks represent a major source of artifacts in the EEG and event-related brain potential. The origin of this artifact is the large difference in potential that exists between the cornea and the retina.
Next is the frequency domain of eye open of the raw data. As you can see from this figure, there are several peaks that indicate by different colors, and we change the time domain to the frequency domain so that we can look and observe several characteristics of the signal that are so hard to look and are not visual at all when we look at the signal in the time domain. So we can see that each signal has different peaks, and the highest peak is the orange color one, and it is indicate C4 or central region.
Okay, lastly, we observe the filter signal for eyes open. As you can see, this is the raw data before and after signal. Before signal, the amplitude is higher, while after filter, the amplitude become lower. So next, we observe the filter between raw data of each electrode in different graph. As you can see, the raw data before signal, there's many highest peak. This is because when we open our eyes, there's a lot of movement in our eyes, so there are much more peaks rather than we close our eyes. So after signal, we can see the peaks become smooth. Alright, next we filter the spectral function. So before we filter, we can see the highest peak is come from the red, which is the C4, while after signal we can see the highest is come from also red, which is C4, and the frequency is like 10 Hz after we filter the signal in spectral function. So that's it from me. Thank you.